This is the 1.2 muscular system process of muscle contraction PowerPoint video. Supporting environment. The stimulus initiates a contraction or reflex arc by a sensory organ noticing or sensing a stimulus. An example of a stimulus could be a tennis ball. The eye would notice the incoming tennis ball hurtling towards it. This triggers a nerve impulse. It's sent from the sensory organ to the central nervous system via sensory neurons. Now, a central nervous system is either the brain or the spinal cord. Once the impulse has been sent through the central nervous system, the impulse then travels through on a motor neuron. The impulse is sent from the central nervous system down the nerves. The impulse reaches the synapse, which is also called the neuromuscular junction. The impulse is sent in the form of neurotransmitters, ACH, across the synapse to the receptor sites. When the impulse has been sent, it travels across the muscle fiber down the two tubules to the sarcoplasmic reticulum, which is just a calcium store. After the sarcoplasmic reticulum receives the impulse, it releases the calcium from its store. When the ATP, which is just the muscle fuel, is broken, it releases energy. Troponin and tropomyosin are covering the actin binding sites. The tropomyosin inhibits the actin in the binding site before the ATP regenerates. The impulse in the T tubules release calcium for the sarcoplasmic reticulum. Calcium binds to the troponin, which causes the tropomyosin to change shape and stop it from inhibiting the actin binding site. ADP is released from the myosin arm, causing it to bind with the actin binding site to form a cross bridge. The myosin pulls the actin along to glide over each other towards the M line. This brings the Z line closer together, causing the muscle to shorten when contracting. The myosin arm will then require more ATP in order to detach from the actin binding site and relax. The ATP impulses need to be continuously sent to the sarcoplasmic reticulum so that the calcium ions are released, which then uncovers the actin binding site. The impulses stop being sent to the sarcoplasmic reticulum, which will stop releasing calcium, which then means the tropomyosin and troponin will return to their original shape and positions, covering up the actin binding sites. A molecule of ATP binds to the myosin arm, allowing it to detach from the actin, which then allows the lines to return to the same distance apart. Now, next to the different types of muscular contractions. There's two types of isotonic contractions, eccentric and concentric. Concentric contraction is where the muscle length gets shorter whilst exerting a force. An example where this could happen in a sport could be in curling a weight and weightlifting. As the biceps brachii contracts to lift the weight, the muscles get shorter to raise the forearm upwards. The eccentric contraction is where the muscle elongates. As the muscle contracts, it gets longer whilst exerting a force. An example of this could be the press up. As the triceps brachii extend to lift and push the body weight against the ground, the muscle gets longer because the elbow extends. An isometric contraction is where the muscle is contracting without the muscle changing length. An example of this could be performing the wall sit. As the quadriceps muscles group contract to maintain the posterior and force applied against the wall, the muscle doesn't move as you stay in one place. An isokinetic contraction is similar to the isotonic contraction, but it's changing the muscle length at a constant speed. An example of an isokinetic contraction would be holding a tennis racket, as the muscles in the forearm must stay gripping the racket so it balances and doesn't fall. So as it contracts at the same rate, the muscle doesn't actually change length.